yeah, the test is going to have 80 questions. And so we have chapter 16, chapter 19, and then gram-positive and gram-negative pathogens. So I'm going to divide it into thirds. So a third of the questions will be from chapter 16, a third of the questions from chapter 19, and a third from gram-positive and gram-negatives. And then from that third will be divided in half gram-positives, half gram-negatives. So it will be pretty much evenly divided. Again, keep track, and there's a chart. There's a chart on eCampus, and instead of a worksheet, there's a chart that a student put together some years ago and then just provided it to everybody else. And it summarizes all of this information. It gives you the diseases, it gives you the prevention, whether there's a vaccine available, etc. So it's a useful chart at summarizing all these. So when we look at gram-negatives, one of the things to start off with gram-negatives is that they're all considered pathogenic or opportunistic pathogens, because all gram-negatives are going to have lipid A. So right off the bat, they're all worse than gram-positives because every one of them is going to have the endotoxin lipid A. So that's for starters. Remember that lipid A is an endotoxin. It triggers fever, vasodilation, inflammation, shock, disseminated intravascular clotting, and it's really the shock that's the worst part, because that means low blood pressure, which can kill the patient. The organisms, gram-negatives, can gain access to the body through some type of mechanical injury in the skin or mucous membranes. The organisms can grow at 37 degrees, which means that they could be, grow in our bodies, and they are pretty good at evading the immune system. So all gram-negatives that can do these can cause disease. So we're going to start a look at gram-negatives by looking at a group of gram-negative cocci. The genus is called the Neisseria genus. And Neisseria are the only gram-negative cocci that causes diseases in humans. The organism is non-motile. They are aerobic organisms, and they're often arranged in groups of 2. They are fastidious organisms, meaning they are difficult to grow. They're picky. They require growth factors. So it's going to be difficult to grow them in the laboratory. It also means that it's going to be difficult for these organisms to grow outside of the body. They don't compete well. They need, again, lots of specialized nutrients. And so that's the good thing, is that they don't live well inside the body. What this means, outside the body. What this means is that they're going to be transmitted by contact transmission, direct contact transmission, because they're not going to do well once they're outside the body. So they have to be transmitted from host to host. We have 2 of the worst of the pathogens of these genus are going to be Neisseria gonorrhoeae and Neisseria meningitides. So we're going to start with Neisseria gonorrhoeae, which causes gonorrhea. It's an STD, sexually transmitted disease. The organism is very good at adhering to epithelial cells of mucous membranes, and it doesn't have to be a mucous membrane of the urogenital tract. It can be any mucous membrane. It's just that it is typically transmitted via sexual contact. The organism typically infects the genital, urinary, and digestive tract of humans, and from there it may spread into deeper tissue and multiply. The organism has become resistant to antibiotics. The prevalence is about 350,000 cases per year, making it second only to chlamydia so it's the second most common STD, second only to chlamydia. Incubation periods 2 to 5 days. Asymptomatic cases occur in both sexes, but it is more common in females. It is an exclusive human pathogen. Okay. The organism is pyogenic, produces a pus-filled discharge, may cause urinary tract infections, and if untreated, it will lead to scar tissue, which leads to sterility. And in males, and we're talking here about males, in males, it causes an infection of the testicles, orchitis. So it could cause infections of the prostate and orchitis. It does show with a pus-filled discharge, and so most of the time it is symptomatic. In females, the organism causes gonococcal urethritis. It is often asymptomatic. The symptoms may be mild pain, maybe a little discharge, but again, females may ignore a discharge, think it is some form of menstrual discharge. They can infect the cervix, other parts of the uterus, including the uterine tubes, and from there it can lead to it can infect the abdominal cavity. It may cause PID, pelvic inflammatory disease. It can result in ectopic pregnancies and sterility. DGI stands for disseminated gonococcal infections. So in this case, we have the organism becoming systemic and is spreading to other parts of the body. The patient may have fever, rash, arthritis, because it often settles in the joints. What makes it pathogenic? First of all, very good at attaching to mucous membrane cells and gaining access to the mucous membrane cell, and from there going into deeper tissue. Once it's in deeper tissue, many of the Neisseria gonorrhoeae are going to be serum-resistant strains. And the serum-resistant strains are going to prevent activation of complement. These more pathogenic strains that become systemic can cause meningitis and endocarditis. So gonorrhea could end up causing meningitis. Okay. Another condition the organism can cause is called ophthalmic neonatorum. This is a condition of babies who are born from a mother who has gonorrhea. So the baby goes through the birth canal and is exposed to gonorrhea, and that may lead to a gonorrhea infection in the eyes that may cause blindness. And this is the reason why all babies, when they're born, they're given a dose of antibiotics on their eyes to prevent ophthalmic neonatorum. A real complication of ophthalmic neonatorum could be if the organism becomes disseminated along other parts of the body, then causes DGI, disseminated gonococcal infections. Again, pathogenicity has to do with the ability of the organism to bind to the host cells, invades mucous membranes, and then from there may go into deeper tissue. The organism is very good at attaching to mucous membranes. Therefore, it has some of those features that allows it to do that, like antigenic variation. So if you remember from chapter 16, we said if an organism had pili and adhesins at the end of the pili, and our immune system cells made antibodies against the adhesins, one of the strategies of the organism was to change the adhesins so that now the antibodies cannot bind them. And so that's called antigenic variation. So now the body has to make new antibodies to attach to the new adhesins. The organism also produces IgA proteases, which destroyed IgA antibody in mucous membranes. OPA is a protein that can attach to T cells, preventing activation of T cells. Factors that can influence incidences of gonorrhea will be the use of birth control pills, because it increases the pH of the vagina and the moisture content of the vagina, making it a more favorable area for infections. The fact that we have asymptomatic carriers and lack of immunity of a population. A diagnosis is typically just by identifying the organism from some type of discharge, vaginal discharge or a discharge from a penile. There are genetic probes, genetic tests that use antigen antibodies. Those are typically expensive though. Treatment, antibiotic, like cephalosporins. Prevention, sexual abstinence, monogamous relationships, correct use of condoms. And that's just showing you the life cycle of the bacteria. It attaches to the mucus, to the epithelial cell of the mucous membrane, From there, it travels to the other side where it's going to be phagocytized by a phagocyte. It leaves the phagocyte and it begins to spread the infection to other cells. The other Neisseria that we're going to look at quickly is going to be Neisseria meningitidis, which causes meningitis. Again, this is a human organism, just like gonorrhea. Humans are the only carriers, the only reservoir. This is an interesting organism because most of us are going to carry, or many of us carry the organism as normal flora of the upper respiratory tract. And it only causes meningitis when the organism manages to breach the blood-brain barrier and go into blood or go into the cerebrospinal fluid. This is the most common cause of meningitis in individuals under 20, so young people. Older people will have already been exposed to the organism because, again, it's a common normal flora, so we're more protected against it. It is transmitted by respiratory droplets or close contact. Again, these organisms, Neisseria, are very weak. They don't live well outside the body. So it's typically, these kinds of meningitis appear in places where there's a lot of people living in close quarters, like a college dorm, for example, or a prison for that matter. The meningitis caused by the organism can be rampant and it can cause death within a matter of hours. So it's a very serious form of meningitis. Okay, signs and symptoms of meningitis. Typically starts as a cold. Following or during the cold, there are throbbing headaches, fever. Next one is very important. Pain and a stiff neck. Stiff neck is a telltale sign of meningitis, extremely serious sign that should really take somebody to the doctor immediately. Sometimes nausea, vomiting, deafness, confusion. Again, that's even worse. Seizures, loss of consciousness. Obviously, these kinds of signs will definitely, urge a visit to the emergency room. Sometimes petechia, which are these purple spots throughout the body due to endotoxic shock. The organism could become systemic and cause blood poisoning. Okay, we do, again, the pathogenicity we talked about already, the ability to attach to mucous membrane cells of the respiratory epithelium. Sometimes they can release endotoxins that causes septic shock and low blood pressure. Sometimes there's a capsule present. In order to diagnose meningitis, they typically do a spinal tap and they retrieve CSF. And they try to culture the organism from the cerebrospinal fluid. Sometimes they take the cerebrospinal fluid through chemistries, and low levels of glucose may be a sign of infection. Okay. Treatment, antibiotics. Typically, intravenous antibiotics is the best treatment. There is a vaccine. It's called the meningococcal vaccine. And it is typically given to children as they enter school, college students as they enter university. So we have 3 organisms so far for which we have vaccines: Streptococcus pneumonia, Mycobacterium tuberculosis, and Neisseria meningitidis. Okay. Next group of organisms is actually a family, not a genus. This is the Enterobacteriaceae family. And this is a group of gram-negative rods. They are facultative anaerobes. This is a huge family with lots of different genera as part of the family. The enteric bacteria, the coliforms, are part of this family. They're typically rods. Sometimes they can be small rods, Coccobacillus. All of them are facultative anaerobes. All of them are glucose fermenters. And this picture is trying to show you some of the features that these Enterobacteriaceae can have. So they can have pillas, through which, and the pilla helps it attach to the host. Adhesins will help it further attach to the host. Iron-binding proteins, hemolysins that destroy red blood cells, flagella, which makes them motile, capsules, which protects them from opsonization. Type 3 secretion systems from chapter 16, which allows them to gain access to the host cell and hide inside the host cell. They have lipid A, an endotoxin. The organisms are identified by culturing the organism using selective and differential media, typically something like EMB. Treatment, antibiotics are usually needed. So oftentimes the condition may be self-limiting, so not even antibiotics may be needed. Okay, now we classify members of the Enterobacteriaceae family into the groups, the coliforms, which ferment lactose and produce gas, non-coliform opportunistic pathogens, and the true pathogens. These are some of the organisms or some of the members of the Enterobacteriaceae family and the conditions they might cause. So E. coli, for example, could cause central nervous system infections. Klebsiella, Enterobacter, E. coli could cause lower respiratory tract infections. Bloodstream infections, E. coli, Klebsiella, Enterobacter. Gastrointestinal tract infections, E. coli, Salmonella, Shigella, Yersinia. Urinary tract infections, E. coli, Proteus, Klebsiella, Morganella. So this gives a rundown of the common infections caused by these organisms, members of the Enterobacteriaceae family. Now, if we look at the coliforms, coliforms are going to be opportunistic pathogens, members of the Enterobacteriaceae family. We know from lab that these organisms are going to be facultative anaerobes, gram-negative rods. We know that they ferment lactose and produce gas and acids. They are a normal flora in the intestines of humans. And the most important member of the coliform group is E. coli. Another important one is Enterobacter. And nowadays, we do have resistant strains of both E. coli and Enterobacter. So E. coli is probably the most famous of all the coliforms. The organism causes gastroenteritis, which is the most common disease associated with E. coli. I think we've all felt it at one point or another. E. coli, right here, this should be in red, is the most common cause of non-nosocomial and nosocomial urinary tract infections. So this is very, very important. The number one cause of UTIs. So the organism is considered an opportunistic pathogen. However, there is a pathogenic E. coli called E. coli 0157H7. That is the strain. Is the most prevalent of the pathogenic E. coli. It causes diarrhea, hemorrhagic colitis, so we're going to have now blood coming out, and more importantly, it can lead to something called hemolytic uremic syndrome, which is an immune disorder that destroys the kidneys, and that is the scary part of the organism. The organism has type 3 secretion systems, which allows it to invade the GI tract epithelium, and they have Shigella-like toxins that have made this organism extremely virulent. So this is an E. coli that has been able to attain the genes for the Shigella toxin, and now it secretes the Shigella toxin, which is a toxin that causes dysentery, very serious type of GI infection. So these E. coli can do the same thing as Shigella. So it's probably the worst of all the E. colis we have. So what you need to remember is E. coli 0157H7 causes hemolytic uremic syndrome, very serious pathogenic E. coli. Most E. coli, however, just cause the normal gastroenteritis, and they are nonetheless the number one cause of urinary tract infections. Among the pathogenic E. coli, we have two, Salmonella, which you probably heard about, and Shigella. Salmonella, both of them actually are going to have type 3 secretion systems, which allows them to gain access to the host. The type 3 secretion system will help it inhibit phagocytosis because it will prevent phagosome-lysosome fusion. It causes the host cell to rupture, to die, and so the organism can free itself from the host cell. These Salmonella is a gram-negative motile bacteria found in the intestines and feces of most mammals, reptiles, birds. Most Salmonella infections in humans are the result of being in contact with animals or consuming infected food. Reptiles, it is a common normal flora in reptiles. So if you have a pet reptile, you should always wash your hand after playing with it. It is also associated with the eggs, with poultry, with chickens. And so we oftentimes get outbreaks of Salmonella due to infected chickens or infected eggs. The last outbreak, which was just a few months ago, was due to fruit that was infected, not even meat. It was probably due to cross-contamination. Two of the Salmonella species are particularly bad. Salmonella typhimurium, which causes salmonellosis, which is typically the kind of Salmonella infections that we see in this country. The other one is a lot more serious. It's called Salmonella typhi, and that one causes a very, very, very severe type of gastrointestinal infection called typhoid fever. So what the organism does is, with its type 3 secretion systems, allows them to go inside the host GI tract lining cell. From there, the organism has been phagocytized. It prevents phagosome-lysosome fusion. So it's just inside the phagocytic vacuole, dividing over and over again until it kills the host cell, and now it's free, and it goes into deeper tissue and gain access to blood vessels, and now begins to spread throughout the body. Salmonella typhi is exclusive to humans. It causes typhoid fever, which is due to consuming food or water that's contaminated with sewage. Obviously, it's not a very common infection in this country, but it is typically of an infection of underdeveloped countries. The bacteria could go from the intestines to the bloodstream, and from there go to other parts of the body, like the liver, the spleen, bone marrow, gallbladder, and become quite serious. Treatment, the use of antimicrobial drugs. However, it has to be careful because you don't want to cause a tremendous release of lipid A. Vaccines are available. So there is a typhoid fever vaccine, which is typically recommended if somebody's going into a country where typhoid fever is prevalent. So that gives us 5 organisms for which we have vaccines. We have Streptococcus pneumonia, Mycobacterium tuberculosis, Neisseria meningitides, and now Salmonella typhi. So that makes 4. Yeah. Okay, so 4 organisms. You need to keep track of those organisms for which we have vaccines. So we're again looking at the members of the Enterobacteriaceae family. We looked at the coliforms, and now we're looking at the true pathogenic members, Salmonella and Shigella. Shigella species, all of them are pathogenic. They're gram-negative, non-motile. So this is the difference between Salmonella and Shigella. Shigella is non-motile, typically found as a parasite of the digestive tract of humans, produces diarrhea, which is caused by an enterotoxin that the organism has. The condition is called dysentery. Sometimes it's also called shigellosis. There are different strains of the bacteria, and all of them cause dysentery or shigellosis. The worst one is Shigella dysentery itself. This organism causes dysentery is hemorrhagic diarrhea. So it causes a complete destruction of the lining of the GI tract with blood, so that everything is shed in the feces, and the feces are bloody. The organism is non-motile, so it manages to get into the host cell. From there, it's going to travel from cell to cell, and it may rupture the host cell, gain access to the blood. And when it's in the blood, it's going to be phagocytized. So it typically doesn't make it into a systemic infection like Salmonella does. Shigella remains a GI tract infection. Okay. Questions about Shigella? All right, so we just have a little bit more to go. The last of the organisms we're going to see is Pseudomonas. and specifically Pseudomonas aeruginosa. Pseudomonas aeruginosa is an opportunistic pathogen. It's a gram-negative rod. It is not a member of the Enterobacteriaceae family. The Enterobacteriaceae family were facultatives. Pseudomonas is an aerobic organism. It is extremely common in the environment, ubiquitous in soil, decaying organic matter. Almost every moist environment is going to have Salmonella, is going to have Pseudomonas. Very problematic in hospitals. It causes, it can cause tremendous infections, nosocomial infections. It is an opportunistic pathogen. So it causes it causes infections, especially in immunocompromised patients. Very rarely is part of the normal microbiota. It causes problems in the lungs of cystic fibrosis patients. The organism causes biofilms among all the pus and the mucus that the patient has. Also causes problems in burn units. Might cause systemic infections in burn patients. Treatment is extremely difficult. The organism is resistant to many antibacterial drugs, and so that's part of the what makes it so scary. It's extremely difficult to combat. Also resistant to disinfectants. If you remember from the lab that we did in class, Pseudomonas was resistant to the antibiotics and resistant to disinfectants. So again, just to recap, Pseudomonas gram-negative, rod, aerobic, which that distinguishes them from the Enterobacteriaceae. Motile, pretty much very common in soil, in water, causes cystic fibrosis, infections in cystic fibrosis patients, burned unit patients, etc. Very resistant to antibiotics. So that is essentially in a nutshell, the importance of Pseudomonas. Okay, questions? Okay. Yes, Jill.
Will the chart help on the test? The chart that you were talking about?
Yes, it will help. I haven't looked at it recently. If it has more organisms than the ones that are currently in our PowerPoints, disregard the extra organisms in the chart, because I used to cover more organisms and I've eliminated them. So just go by the organisms that are in the current PowerPoints. So yeah, it will definitely help you. So expect things like, a matching of organisms with diseases that they cause. Know which organisms we have vaccines for. A little bit of the virulence factors. We talked about protein A, protein M protein, both in chapter 16 and in this chapter. So you have to have an idea of that, because we talked about them in both chapters. Type 3 secretion systems and Salmonella. We talk about that in chapter 16. So all of these repetitive themes that we've had from both chapter 16 and this chapter, you should pay attention to those themes that repeat themselves. Serum-resistant Neisseria, we talked about those on chapter 16. We talked about Streptococcus having the hyaluronic acid capsule, having the capsule that deactivates C3b to prevent opsonization. Okay, so those are the kinds of material that is found in both chapter 16 and gram-positive and gram-negative chapters. So pay special attention to those. Any other questions? Yes.
And what is the percent of this grade on this one, Ms. Sarah?
You're talking about this material on the, I'm not sure I understand. You're talking about the lecture test 6? Oh, same as the other tests. But this is the extra test that will be will take the place of the lowest lecture grade. So in other words, if you're happy with your grade up till now, then you don't have to take this test number 6. You still have to do the quizzes for the module, but you don't have to take the test, because then this will be the drop test. If you think that you could do well on this test and that grade take the place of your lowest lecture test, then you should give it a shot. Yeah.
Let's say you have a C, barely a C, maybe like, around there. Would it be a good idea to take this test even though you're not so sure about it?
You might want to wait until the practical and see how you do in the practical and then decide. The unknown grade typically helps the students. The unknown grade is not going to hurt you. It's on the contrary. Most of the students do very well in the unknown, and that helps. So you don't have to worry about that. At this point, the one that you might have to worry about will be the second practical, which in general is finding that students do better on the second practical than they do in the first practical. So if you wanted to wait until then to decide whether you're going to take the test or not, you can do that. And if it doesn't help you to take the test, then you don't have to take it. Again, you still have to complete the quizzes and the discussion questions, but you won't need to take the test. So you may want to wait until after Tuesday to decide whether this test will help you or not.
I have a question.
Yeah.
So I didn't take test 3, so that was like the test